Hello, and welcome to the scientific exploration video that takes place in Point Pleasant Park, situated on the southwest coast of Halifax, Nova Scotia. Today, we are focusing our attention on the intriguing species known as Semibalanus balanoides. To begin, barnacles belong to the class Crustacea. These fascinating creatures possess calcareous shells, providing them with protection and structural support, essential for their survival in the challenging intertidal zone. Barnacles, including Semibalanus balanoides, exhibit a global distribution, occupying diverse coastal regions around the world. In this study, we direct our attention to the barnacles thriving along the rocky shores of the Atlantic coastlines. Despite their seemingly simple lifestyle, barnacles possess intriguing reproductive strategies. Each individual possesses both male and female reproductive organs. To avoid self-fertilization, barnacles cross-fertilize with neighboring individuals to enhance genetic diversity within their populations. The barnacle larvae undergo a series of transformations, shedding their swimming appendages and settling on suitable substrates. Once settled, they undergo metamorphosis, permanently attaching themselves to substrates, thus entering their adult life stage. Today, we'll be discussing the settlement patterns of barnacles. These organisms display a preference for hard substrates, such as rocks, ship hulls, and piers. This scientific experiment takes place in Point Pleasant Park. Because of this, I would like to acknowledge the significance of this land, which forms part of the ancestral territories of the Mi'kmaq people, and I honor their ongoing connection to this place. My study focuses on the relationship between rock sizes and barnacle settlement, specifically looking at the percent cover of barnacles on three distinct size groups. I conducted the field experiment at low tide by employing a 1 meter squared quadrats that were systematically distributed along the transects within the study area. The study involved categorizing the rocks into three distinct size groups: small rocks, less than 30 centimeters, medium rocks, 31 centimeters to 90 centimeters, and large rocks, greater than 91 centimeters. Within each size category, barnacle settlements were assessed using four quadrats, ensuring representative sampling across the study area. Now, going to our methods. Before going to Point Pleasant Park, a lot of preparation was needed. I packed my backpack with the necessary supplies, including snacks to keep me energized, a phone for communication and documentation, a first aid kit for any unexpected situations, and my data sheets to record my observations. Once I arrived at my designated field site, I carefully assessed the area. It was crucial to choose a location that offered a variety of rock sizes, as well as a distribution that spanned the entire intertidal zone. This ensured that my experiment captured the full range of conditions present in this dynamic habitat. To systematically collect the data, I established transects along the intertidal zone, ideally parallel to the low tide mark. Using a measuring tape, I carefully measured the distance from the highest intertidal depth where barnacles were present to the low tide mark. This allowed me to determine the location for each transect. With the transects in place, I moved on to the meat and potatoes of my experiment, the quadrats. Using a random number generator, I selected points along each transect to place my quadrats. I repeated this process until I collected data from four quadrats for each rock size category. As I examined each quadrat, I carefully documented the percent cover of barnacles on the rocks. I did this by capturing a photo of each quadrat so that I have a permanent record of all my data. I also recorded approximations of the percent covers on my data sheets while I was at the experiment location. After collecting my field data, it was time for the analysis. I collected the average percent cover for the four quadrats of each rock size category. To explore the significance of my findings, I performed a two-way analysis of variance, or an ANOVA, without replication. This allowed me to assess the variance within each rock size category and between different rock sizes. Then, to visualize my results, I created a bar graph. As you see, the y-axis represents the percent cover, while the x-axis represents how far down the intertidal zone the quadrats were placed. Then on the far right, I have the averages for all the quadrats of each rock size category. Moving on to the results. Um, Now that I have collected the data and conducted the analysis, let's go into the results. The average percent cover of barnacles on the different rock sizes was as follows. For large rocks, it was 0.144. For medium rocks, it was 0.037. And for small rocks, it was 0.004. Those are all percentages, by the way. Um, to gain a deeper understanding of the data, I examined the variances within each rock size category. The variance for small rocks was 0.000002. For medium rocks, it was 0.001638. And for large rocks, it was 0.007225. Additionally, I calculated the variance of the average percent cover, which was found to be 0.00535. The results revealed a interesting trend. As we move to the lower intertidal depths, the average percent cover of the barnacles on the rocks increased throughout the sizes. This indicated that barnacles have a preference for settling on larger rocks as we move towards the lower intertidal zone. Moving on to the statistical significance of my findings, the p-value obtained from the ANOVA was 0.00847. This suggests that rock size has a significant effect on the percent cover of barnacles. In other words, there's a strong relationship between the size of the rock and the amount of barnacle cover observed. These findings support my hypothesis that larger rocks would have a higher percent cover of barnacles compared to smaller rocks. Now on to the discussion. Looking back at the bar graph, it clearly illustrates the significant difference in barnacle percent cover among different rock sizes. Large rocks exhibit a substantially higher percent cover compared to the medium and small rock sizes. The statistical analysis further supports this observation, as the obtained p-value confirms the significance of rock size in influencing barnacle percent cover. To support my claim, a article completed by Hills and Thomason in 1998 stated that in their study of barnacle densities on different sizes and surface textures of tiles, they found that barnacles, semibalinous balanoides, had a significantly higher settlement rate on larger tiles. The largest tile, 20 centimeters squared, sequestered the largest density of barnacles compared to the other tiles of differing sizes. Unfortunately, my study does not definitively prove anything, and I must acknowledge the potential sources of error and limitations in my study. Um, the size of quadrats used for my data collection may have posed some limitations, as it restricted the scale of my observations. Additionally, the number of data samples collected could also impact the overall accuracy of my findings. Moreover, the study was conducted during a specific time of the year, which might have influenced the results. The range of data was limited to a small experiment site. Also, factors like pollution and high disturbances could have introduced variability as well. To address these limitations, I'd like to address the potential avenues for future research. First, conducting the experiment with a range of quadrant sizes would provide a more comprehensive analysis of barnacle settlement patterns. Additionally, exploring multiple locations and conducting experiments at different times of the year would offer a more holistic view. Lastly, selecting a different barnacle species as a target organism could provide valuable insights into species specific settlement preferences. The implications of my findings extend beyond scientific curiosity. Gooseneck barnacles, being a potential food source for humans, hold promise in the culinary world. With their position low on the food chain, their inclusion in aquaculture systems could enhance diversity and contribute to more sustainable practices. Polyculture systems incorporating gooseneck barnacles have the potential to improve the overall sustainability of aquaculture. In conclusion, the study focused on the settlement patterns of barnacles and the relationship with rock size in Point Pleasant Park. The results demonstrated a clear association between rock size and the percent coverage of barnacles. The data revealed that large rocks exhibited significantly higher percent coverage of barnacles compared to the medium and small rock sizes. These findings align with previous research done by Hills and Thomason in 1998, which also observed a higher settlement rate on of barnacles on larger surfaces, specifically the 20 centimeter squared tile size, sequestered the largest percent cover. Um, Although my data did reveal a significance, it is important to acknowledge the limitations, potential sources of error in my study. The size of the quadrats and the number of data samples collected may have influenced the precision of my findings. The time of the year limitation, range of data across the experiment site, and factors such as pollution and high disturbance areas could have introduced variability into the results. Future research should be considered using a wide range of quadrant sizes to capture a more comprehensive view of barnacle sediment patterns. Conducting experiments across multiple locations and throughout different times of the year would enhance the generalizability of the findings. Exploring different barnacle species as target organisms could provide additional insight into their settlement preferences. Finally, my study's findings shed light on the intricate relationship between rock sizes and barnacle settlement, offering potential insights for more sustainable aquaculture and ecological conservation. And now I would like to thank you all for watching my scientific exploration video. Have a wonderful rest of the day. Thank you.